So I'm going to tell you a little bit about a long non-coding RNA, and I put that in the context of cardiac disease. So it's not a course about cardiac disease, but I'd like to give you two slides of introduction to put everything in context again. So we're interested in cardiovascular disease, which is still a major problem in terms of morbidity and mortality in the world, and particularly in Western countries. And the focus of our research is really the adaptation of the heart to stress. And a stress in the heart can be what's happening after myocardial infarction. So coronary artery occlusion, then you have tissue damage that destroy the tissue, and obviously the heart has to adapt. Now there's players that are very important in this tissue repair mechanism that take place in the heart after damage. And you have fibroblasts that generate a matrix and a fibrotic scar. You have the cardiomyocyte, and the cardiomyocytes are the muscle cells of the heart. So the fibroblasts mostly proliferate. The cardiomyocyte adapt via hypertrophy. They don't proliferate. And then you have some cells that are not completely defined, that are cardiac precursor and immature cardiomyocyte that can still provide a source for generating a new tissue, a new muscle, I would say. And in the heart, you end up with this fibrotic scar that is very detrimental in terms, again, of mortality and morbidity. So the whole idea is to find ways to tip the balance towards something more regenerative. So this is the focus of our research, to try to recruit the appropriate cell types, mostly cardiac precursor, immature cardiomyocyte, to create a new muscle in this damaged heart. Okay? That's the extent of what I'm going to tell you about the heart. Now, everything that is all this complex process, biological process, are controlled by what we call the cardiac gene regulatory network. And this network controls gene expression, coding gene expression, mRNA production. And this is under control by cardiac transcription factor that in a combinatorial manner to control the expression of these coding genes. But more and more, we appreciate the importance of non-coding transcript, non-coding RNA. And with this, we have a focus on long non-coding RNA that are defined as transcripts that are larger than 200 nucleotides in size. And among also this non-coding RNA groups, you find a particular type of RNA that are associated to enhancer, that we call enhancer RNA. And you know about microRNA. That is not the focus of my talk today. And all this control various processes in organs. So this is example of the heart, but this control developmental and cardiac morphogenesis, the cardiac differentiation in vitro, for instance, and when you try to differentiate embryonic stem cells or induced pluripotent stem cells into a given cell type, and also adaptation of the adult organ to environmental cues, right? Such as stress, myocardial infarction. So the importance of the non-coding genome is now more and more recognized. And this is a slide that you might have seen already. But the main message here is that the main difference between mammals and other species with a more compact genome, the main difference is the amount of the non-coding genome. So complexity is not really associated to the number of coding genes, but to the amount of the non-coding genome. And among this non-coding world, we have known RNA that has been known for quite a long time, microRNA, piRNA, and so on. But you have this group of long non-coding RNA that contribute a lot to this number of non-coding genes. So how do you classify, annotate this non-coding, long non-coding RNA? And you can use different ways of annotating classify. First of all, you can consider the context, the genomic context, and you can find transcripts that are generated by purely intergenic genes that are non-overlapping with coding genes as compared to coding genes. So you have overlapping transcript. They can be intronic, exonic. They can be sense, antisense. These are the most difficult group to study. And you can have also divergent transcripts that are transcribed in the other direction as the coding gene. So obviously this one are the easiest to study. Now you can also study the chromatin context, and you can try to define whether this transcript comes from a region that are associated with particular chromatin mark that define, for instance, enhancer or promoter. I come back to this in a minute. So, for instance, this H3K4 monomethylation enrichment define enhancer element, or putative element in the genome. And this is more associated to promoter. Now you can find, or you can also consider subcellular localization, whether the transcript are mostly nuclear or also cytoplasmic. And this gives information regarding whether the transcript is involved in more, let's say, cis function versus trans function. Okay? And you can consider the structure, whether this transcript can associate to other RNA, to DNA, to RNA binding proteins. And you can consider the function that you can define different ways. So the mechanism of action of long non-coding RNA has been defined in different fields, in different experiments. But mostly what link RNA do is they associate to chromatin modifier. And they give a non-specific system, such as the associate to the chromatin modification, a specificity. So they provide some kind of address code to a non-specific system to target this modifier to specific location in the genome. So they can act as signals, so that modifier change modify the genome at particular location. They can act as guide or scaffold for this different protein complex, and then regulate gene expression. They can also act as decoy, for instance, for splicing factor. So they associate, and then the splice factor cannot find its target. Also associate to microRNA, and then titrate this microRNA away from the natural target, and then increase gene expression, coding gene expression this way. And one important group, again, are the one associated to enhancer. And the idea here is that this transcript promotes the formation and stabilization of the loop between the enhancer sequence and the promoter sequence. I'll come back to that. Now, this is an example of the taken from the cardiovascular field, but there's more and more of these transcripts that are known to have real function in the organ, in the heart, in this particular instance. So this is a list of link RNA that has been fully and functionally characterized. Okay. So you see that they are involved in metabolism, in controlling hypertrophy of cardiomyocyte. They are associated to the conduction system in the heart. They can also be important for differentiation, proliferation during development. But in the heart, there's also different types than cardiomyocyte, different cell types than cardiomyocyte. There's fibroblasts, inflammatory cells, endothelial cells, smooth muscle cells. And for all these instances, we have some transcript, long non-coding RNA transcript that have been shown to be functional. Now, just a quick word about enhancer. So enhancer, as you know, are regulatory units that instruct gene expression, spatial and temporal gene expression. Now, they're very important for defining cell identity and during development and disease. And you can annotate enhancer based on different parameters. Hypersensitivity to DNAs, they are associated to a higher ratio of H3, K4 monomethylation versus H3, K4 trimethylation. When they are active, they are also associated to H3K27 acetylation. They bind co-activators such as P300, and they bind multiple transcription factors. Okay? Now, there's a great number of enhancers in the human genome, but probably 1 million. So this is tremendously important if you compare to the number of coding genes, for instance. That's around 20,000, right? And so far, we have identified approximately 50,000 of those that produce or that are associated to a transcript generated from these particular locations. So if you consider only this group, they are characterized by higher binding of co-activators, a greater enrichment of histone marks, binding of lineage-determining transcription factors, and also this correlation with the formation of this promoter enhancer loop. That's important for the activation of gene expression, of coding gene expression. So eRNA are important for regulating chromatin accessibility, the binding of the RNA pol II, and the formation stabilization of a loop. And eRNA can exert cis and trans function. So this is taken from a very recent review, and I like this classification because not every enhancer can produce a functional transcript. This is still hypothetical for most of them. So this group, the Rosenfeld group, considers 3 classes. So in class 1, really the transcription is at the enhancer is kind of, the transcript at the enhancer is kind of a byproduct of transcription. So you do have the transcription machinery that is closely associated to an activated gene. And because you have the machinery there, you produce a transcript. It doesn't mean that the transcript is functional. Okay? That would be one situation. And the machinery can associate primarily to the enhancer and then to the promoter, or primarily to the promoter and then transcribe the enhancer. So it works both ways. Here you have a role for the transcript and the enhancer, which is to bring the machinery in the close vicinity to the promoter of the gene you want to activate. And you promote this induction by remodeling the chromatin. It can also interfere with transcription by promoting transcription in the other directions than the gene. And this is the truly functional case, right? Class 3, in which the transcript that is generated from an enhancer can promote this association between the promoter and the enhancer, can also have a trans function by associating with gene on a different chromosome, for instance, or to have true trans effect, because the transcript will travel, possibly associating with RNA binding protein, chromatin modifier again, and have a different function than this cis function, and can act as a decoy as well. So this is taken from the same review that I've just mentioned, and I think I like this classification. So this is eRNA, the eRNA world. So that transcripts are generated bidirectionally from an enhancer sequence. This is let's say, the benchmark situation. Then you have the link RNA groups that are characterized by transcripts that are very much resemble a messenger RNA. They are capped, they are polyadenylated, they are multi-exonic, and these two groups are characterized by very high tissue specificity, cell specificity as well. Now, what the author have considered is that in some instances, enhancers are associated with link RNA, not with just bidirectional transcript. And this small group here, we believe, are extremely important in controlling cell identity. And by chance, this is the group we have been studied in the context of our research. So this kind of summarizes maybe the situation. So this is an enhancer, promoter, and so one situation, classical cis activation, when you have the enhancer that associated to the promoter via a loop, and the transcript is important for formation and stabilization of the loop, and then activate the gene. Then you can have kind of a cis activation via transregional association, as I call it. So the enhancer will associate to a promoter on a different chromosome, for instance. But these two situations are very much alike. Okay, so this would be also kind of a pseudo-cis. Then you can have a trans activation because the transcript travels in the genome, find its target, and then activate the target genes. But you can have all the transfunction association with RNA binding protein, chromatin modifier, decoy, etc. Now, another concept that is emerging is the concept of super enhancer. A super enhancer are a cluster of active enhancers. So when you have enhancers that are close by, and they are stitched, then you have what we call a super enhancer. So these again are marks that define an active enhancer, particularly, let's say, the K27 acetylation. So this would be a situation when you have several enhancers that are clustered together, and this would be defined as a super enhancer. Okay. Now, if you want to identify a super enhancer, sorry, what you do, you actually study the enrichment of particular region of the genome for lineage determining transcription factor, for instance, or the association with particular chromatin mark or other co-activators. And you rank all these enhancers from low to high, and you create this kind of a hockey stick curve here. And then you consider the inflection point where the slope is equal to 1. And whatever is on the right side here is called super enhancer. And the rest is typical enhancer. Okay. So you see that you have only 1% that are defined using this particular criteria. 1% of enhancer are super enhancer. But they're extremely important for controlling cell identity. So this is, again, hypothetical, but I mean, this could be a super enhancer that is only active in cardiomyocyte and control the identity of the cardiomyocyte. And this super enhancer wouldn't be active in another cell type, such as the fibroblast, but you have another one that is active in this case, that controls the identity of the fibroblast. Now, these super enhancers are apparently very important in development and disease. And this is a very important study in which the author have used GWAS study, genome-wide association study. and consider about 5,000 SNPs that affect, that are associated with disease. And this is the percentage of each SNP that are associated with each disease. The important message here is that more than 90% of these SNP are in non-coding region of the genome. They don't affect coding genes. They affect the non-coding gene. But more importantly, if you consider now the disease and the SNP that are associated to disease, they are located in enhancer and super enhancer that are active only in the tissue of interest. So if you consider, let's say, Alzheimer's disease, they are more likely to be associated to a super enhancer that are active in the brain. If you consider, for instance, trait, electrocardiographic traits, the SNPs that affect this, who are associated with this trend, are more likely to be located in super enhancer that are active only in the heart. Okay, so with this, I'm going to give you a little bit of information on how we approach the study and characterization, identification and characterization of the long non-coding RNA in the heart. But it can be applied to any tissue. What we do is that obviously we have biological samples. We isolate the poly-A RNA fraction. This was a choice at the beginning just to facilitate the characterization. And we perform very deep RNA sequencing. I'll come back to this in a minute, but we perform 400 million, we generate 400 million reads per sample, which is extremely high as compared to what people use usually, which is in the range of the 50 million. Then we use a pipeline to reconstruct the transcriptome. So that is just to identify the transcript. And from this transcript, we can separate this transcript into what is known. And in this group, we have the messenger RNA. But we have also the annotated long non-coding RNA, identified by others. If it's a novel transcript, then we check the coding potential. And if there's no apparent coding potential, then we consider transcripts that are higher or greater than 200 nucleotides inside. Then we call them novel long non-coding RNA. Okay. Now, just a word about the depth of sequencing. This is what we use. I mean, this is from two of our studies. So this is the number of novel long non-coding RNA that we have discovered. And this is the sequencing depth. So it's a retrospective study. So this is basically the transcript that we have discovered that we wouldn't have discovered if we would have used the 50 million read. So you see that we would have missed about 75% of the transcript. Now the reason is that the transcript, the long non-coding RNA transcript, are very lowly expressed. So since the abundance is low, is very low, then in order to reconstruct the transcriptome, you have to accumulate reads. And to accumulate reads, you need to sequence your samples at a very high depth. Okay? We think that this is very important because if you're interested in enhancer RNA, for instance, then you want also try to detect transcripts that are important as nation transcript. So they might have only a few copies per cell. And this is the only way you can detect it. It's very demanding in terms of, because it costs money. It's also demanding in terms of bioinformatics, but it's extremely useful to identify relevant transcripts. Okay, with this, how do you characterize the novel long non-coding RNA. So again, this can be applied to any tissue. So this is the part I just described when we identify novel long non-coding RNA. But then we have tried to functionally characterize this transcript. We have considered the organ specificity. We have correlated this with parameters of physiological parameters, association with chromatin marks, and other characteristics that I won't discuss today. And very important, we also try immediately to make sure that we have autologs in humans in order to identify relevant target for the clinics. This is because we're very much interested in finding ways of improving the adaptation of the heart to stress, and we put everything in a clinical context. So in this particular study, when we have compared myocardial infarcted heart, this is in the mouse, versus a normal heart, we have identified about 1,500 novel long non-coding RNA. So this is from a single study, so this is a lot. Now, about 25% of those are modulated after infarction. Okay? They have no coding potential, and this is just to make sure that all samples are very homogeneous. So as you can see that these samples clustered together, it means that the transcriptome reacts to stress in a similar fashion from one sample to the other. So this, gives support to the fact that this non-coding transcriptome has physiological function in a stressed organ. So the first thing we try to do is to find whether this transcript were more tissue specific or than other transcript. And we use publicly available database, the ENCODE data in this case, to check the expression in a variety of non-cardiac tissue, 17 in this case. And we compare the expression with expression in the heart. So for each transcript, we define a score. And if you see a red dot here, it means that the transcript is heart-specific. And we perform the analysis systematically for all transcripts identified, including the messenger RNA. So you see that you have a few that are heart-specific. But if you consider now the long non-coding RNA group, the novel ones, you see that this is very much increased. And this is the quantification, and this is a very striking finding. You see that if you consider the differentially expressed transcript, 60% of this transcript are exclusively expressed in the heart. Very, very high specificity. In trying to define the function of this transcript, we try to associate this with parameters of cardiac function and remodeling. So it's not important that you know what is in there, but I mean, you have to know that you have parameters that are mostly associated to remodeling and parameters that are mostly associated to cardiac function. And again, we try to cluster this transcript according to the correlation with these different parameters. And what you can see here, that you can immediately find different clusters. Cluster 2, for instance, is closely positively correlated with parameters of remodeling, whereas cluster 4 is positively correlated with cardiac function. It means that you can now have transcript that control different processes in the heart that are normally associated, but now you can segregate these different parameters and identify marker and potentially functional transcript that define these parameters separately. So it's even more striking when you look at individual parameters. So this is ejection fraction, this is the function, this is the size of the infarct, this is the thickness of the wall of the septum in this case, this is the diameter of the ventricle. So they all correlate with cluster 2 and cluster 4. But if you look at the individual parameter, you see that, for instance, this thickness of the septum is also correlate with cluster 1. So now in this small group, you have a couple of transcripts, maybe more, that define uniquely this parameter. It is a fantastic biomarker for following the remodeling in the heart of particular region of the heart. Now, in our field, this is extremely important if you want to apply the appropriate therapy, targeting potentially tissue remodeling or sustaining function, or possibly targeting the conduction system, for instance. Now, this is an example of a study of a characterization when we used also publicly available database. In this case, we used the chromatin context in order to define each transcript. So this is an example in which we checked different marks that are that are present or not in different organs. And as you can see here, in this particular case, you have this association with H3K27 acetylation mark, and uniquely in the heart for this particular transcript. It means that in the heart, this transcript is associated with an active enhancer. Now you can do a systematic analysis for each transcript again. So now you have these different groups, the messenger RNA, the known long non-coding RNA, and the novel long non-coding RNA. And you can consider only this purple block here. What you might immediately see is that you have a bigger association with enhancer for long non-coding RNA, novel long non-coding RNA than for the other two groups. This is clearly evident for the heart, but it's also true for the other organs. So novel long non-coding RNA are more likely to be associated with enhancer. This is also true when you assess differentiation. So this is an experiment in which we have differentiated ES cell into cardiomyocyte using a particular method that we call the hanging drop. And you can define different stage, embryonic stem cell stage, the mesodermal stage, the cardiac precursor stage, and the cardiomyocyte. And again, you see that if you consider this purple block here, the novel long non-coding RNA are closely associated to enhancers that are active, particularly at the mesoderm, cardiac precursor, and cardiomyocyte stage. What is very important, you can also analyze this dynamically, and you can define enhancer associated links that are only active at particular stage, not during different stage, I mean the subsequent stage of differentiation. I mean, this is a more evident situation in which you have active enhancer that are only active in cardiomyocyte. So these are obviously very important for controlling cell identity, and this could be targeted for different purposes. Okay, so this is a more difficult situation, but I think it's an important piece of information. Now, if you try to define function of a transcript, you cannot rely on a simple expression at different stage. So again, you have this differentiation process, and you have, in this situation, 3 genes that are co-expressed. Okay. This doesn't tell you anything about the function of these genes. This could be a transcription factor, this could be a contractile protein, or whatever. No? We're talking coding gene now. So what this author have demonstrated, it's a very important paper, that if you consider the chromatin mark that are associated at each gene during differentiation, then you can define patterns. So they used these 4 chromatin marks. There's 4 different stages of differentiation, and they come up with 34 patterns of chromatin state transition. Now with this, they can obviously group the genes into 34 different clusters, 34, and what they have found is that now these clusters are associated with particular function. So this is the list. It's not too important, but let's just consider these 3, for instance. Cluster 1, 2, 3 are more associated with housekeeping function. What is very interesting for us is that in this cluster 20, 23 to 27, you have all the genes that define contraction and heart development. So we thought that we could use this information and try to perform the same analysis for the transcript we have identified, and then group the long non-coding RNA transcript into different clusters. And since we know about the function that is associated to each cluster, we can define a function for the long non-coding RNA transcript. So it's exactly what we did. And so this is what we have found for the messenger RNA. You see that a large proportion of the transcript are associated with housekeeping function. The situation is very different for the novel long non-coding RNA. Now, they fall into this cluster that define function in the heart and development of the heart. And this really support a function for this long non-coding RNA transcript. This summarizes a little bit what we have find for one particular transcript that we call novel link 6. It's not really a good one. We should have we should have used another name. So this transcript is downregulate after infarction in the border zone of the infarct or in the remote zone of the infarct. It's differentially modulate. It's positively correlate with function. This is a very high correlation, actually. It's associated with an enhancer sequence, an enhancer element. We know that this is an enhancer element because we can take this piece of DNA, generate a transgenic animal, and target reported gene expression in the heart. It is also present in cluster 25, together with a cardiac gene, BMP10, that control NKX 2.5, which is an important cardiac transcription factor. And if you now use oligo to downregulate the expression of this transcript in cardiomyocyte, you have this downregulation here. You modulate BMP10 and also NKX 2.5, but you don't touch other important cardiac genes, suggesting high specificity in the control of this gene expression. And importantly, we can also find a situation in humans in which this particular transcript is modulated. We have considered aortic stenosis, in which function is maintained, and dilated cardiomyopathy, in which function is decreased. And you can see that in this situation, novel link 6 of the ortholog, the human ortholog, is downregulated, and we even detect downregulation of NKX 2.5, which is clearly relevant. I'm not sure how much time I have, because
you have 10 minutes, but you use some time for discussion.
Oh.
So you say,
yeah, okay.
30 minutes?
Okay, maybe I'll skip a part in the middle, but I'm going to discuss this one. So, regarding the importance of enhancer-associated RNA, in order to test the reality during development, we have considered bona fide enhancer that have been identified using enrichment for P300. So these are the enrichment at particular putative enhancer. These are the transgenic experiment in which we take this DNA element and we can target reporter gene expression in the heart. So we know that they are true enhancer. But now what we consider is the transcription of this enhancer. And as you can see, you can detect a transcript for each one of them, only in the heart, not in other part of the body during development. And these enhancers are transcribed in a dynamic fashion during development. So these are staged during development. Sorry if you cannot read that, but this is E11 or E9.5, sorry, and this is P0 and P30. So you see that it's dynamically transcribed for all of them. And what is more interesting is that the adjacent gene, which are very important cardiac gene during development, are also dynamically transcribed, and you have a clear correlation between the transcription of the enhancer and the transcription of the cognate coding genes. This is also true during differentiation in vitro. You see that you have activation of this transcript, correlation with the adjacent genes. This is quite evident. What is important is that acquisition of active mark for this enhancer correlate with activation of the coding genes also. And this occur at very defined stage of differentiation, particularly at the cardiac precursor stage in this case. As you can see that you have also activation of the coding genes at a similar stage. And this is the ultimate experiment in which you, to test the function, you have to use knockdown experiment of the transcript in order to prove that this transcript have a function. You cannot mutate the DNA element, because if you mutate the DNA element, it doesn't tell you that the transcript is important, right? So we use a short hairpin in this case to target this MM85 transcript. And what you see immediately that you can downregulate this one, and the cognate genes, myocardium in this case, is downregulated as well. And this is actually a gene that is closely associated to the enhancer, even closer in terms of location in the genome, but it's not affected. So it's very specific. Okay, so if I have a few, maybe I skip a part of this part. Maybe we can discuss at the end. And I finish with this. So here, in this case, we try to refine a little bit our analysis. So we started again from the study I described using the myocardial infarction model in the mouse. We consider only purely intergenic London coding RNA, okay, in this case. That's why the group is a little bit lower. The number is a bit lower. We select heart-enriched London coding RNA. We consider only the one that have human orthologs. And we consider now the one associated to super enhancer. And with this, we have only 6 in this group. Then we check the specificity, the cell specificity, cardiac fibroblast versus cardiomyocyte. And only one was clearly enriched in cardiac fibroblast as compared to cardiomyocyte. So for us, this is a very important target because modulating cardiac fibrosis in the heart is a challenge. We don't have any specific target to go only to the cardiac fibroblast population and modulate fibrosis, which is detrimental to heart function. Very relevant clinical target. Now, what you see here is again okay, we have called this gene WHISPR because it's in the WISP2 locus. Okay, this is anecdotal. So you see that it is highly expressed in the heart as compared to other tissue. But what is more important, that if you consider different source of fibroblasts, we have here the cardiac fibroblast obtained from neonatal mice or from adult mice. And tail fibroblast, lung fibroblast, or embryonic fibroblast. You see that the transcript is highly enriched only in cardiac fibroblast. So with this, we can differentiate between the source of fibroblast in different tissue. If you consider the usual marker of fibroblast, this is absolutely not the case. So this is really to emphasize the tremendous specificity of this transcript. This is a unique target. Then we checked cytoplasmic versus nuclear expression. As you can see here, WHISPR is expressed in both compartments, suggesting that it might have cis function, but also trans function. WHISPR is dynamically induced after infarction. It's by in situ is clearly enriched in the remote zone. Sorry, it's the border zone. Sorry, this is clearly a mistake. It's the border zone of the infarct. You can modulate now WHISPR expression using gapmer, this oligos that target transcript using the RNase H mechanism. You see that you have downregulation of a number of genes that define the identity of the cardiac fibroblast. You can modulate proliferation, fibroblast migration, fibroblast apoptosis. If you downregulate this transcript, basically you affect the whole function of the fibroblast, the identity of the function of the fibroblast. Then you can use this gapmer in vivo after an infarction in the mouse. So we can inject this gapmer 2 days after infarction, very relevant window in time for in a clinical setting. Again, we downregulate WHISPR. We have the relevant downregulation of the fibroblast gene in the heart. We basically don't touch much the marker of cardiac cardiomyocyte, but we have downregulation of this marker of stress, which suggests that we create beneficial effect in this heart. Indeed, with this, we completely reduce the size of the infarct. Okay, this is an infarcted heart, and this is the one treated with the gapmer. As you can clearly see, the size of the scar is very much decreased. The interstitial fibrosis is also decreased. And more importantly, we have improved function. So this is again the infarct size that is reduced, the remodeling is reduced, and the function is improved by targeting a single long non-coding RNA transcript that targets a single subpopulation in the heart. And this is again the human data. We can detect now WHISPR in the heart of this patient here. These are aortic stenosis patients. You see that the collagen volume is different between these two cohorts, so it's why we selected those. So you have severe fibrosis in one case, non-severe fibrosis in another case, and the transcript is directly correlated to the amount of fibrosis. And again, specificity here. Human cardiac fibroblasts do express WHISPR versus human dermal fibroblasts do not induce WHISPR, suggesting that we can now use this target for modulating fibrosis in the heart of human patients. So this kind of summary of my talk, I think we have now in lincRNA a group of molecules, and particularly in lincRNA that are associated to enhancer, a group of molecules that are tremendously important for controlling function, or cell function, organ function, and could be a fantastic target for therapy. You can even imagine to modulate now directly this transcription or directly in organs in order to modulate cell behavior and organ behavior. So with this, I'm going to stop. I'd like to thank all the people in my group that have been involved in this work, and our collaborator outside as well as in Lausanne. Thank you very much.